Advances in gene modification, unlocking the potential of gene therapy. Title: Advances in gene modification, unlocking the potential of gene therapy. Introduction. In recent years, the field of gene therapy has seen remarkable advancements, breaking new ground in the understanding and treatment of genetic diseases. Gene modification, a key component of gene therapy, holds the potential to revolutionize medicine by targeting the root causes of various disorders. This article explores the latest advances in gene modification and their immense potential in unlocking the doors to effective gene therapy. Understanding gene modification. Gene modification involves manipulating the genetic material of an organism to introduce desired changes. This process can be achieved by several techniques, including gene editing, gene addition, and gene silencing. By modifying specific genes, researchers can correct genetic defects, enhance the body's natural defenses, or address disease pathways at a molecular level. Gene editing techniques. One of the most promising gene modification techniques is CRISPR-Cas9, a revolutionary tool that allows scientists to precisely edit genes. CRISPR-Cas9 acts as molecular scissors, targeting specific genes and introducing modifications. This technique has sparked immense excitement due to its simplicity, cost-effectiveness, and high efficiency. With CRISPR-Cas9, the potential to correct genetic defects associated with diseases like cystic fibrosis or sickle cell anemia is within reach. By specifically targeting and modifying problematic genes, researchers aim to offer potential cures or at least reduce symptoms significantly. Although challenges remain, such as ensuring accurate edits and minimizing off-target effects, the possibilities offered by CRISPR-Cas9 are transformative. Gene addition approaches. In addition to gene editing, gene addition approaches have shown great promise. This technique involves introducing healthy genes into a patient's cells to compensate for defective or missing ones. Viral vectors, such as adeno-associated viruses, are commonly used to deliver the therapeutic genes into the target cells. Gene addition has shown encouraging results in treating genetic disorders like spinal muscular atrophy, SMA, and hemophilia. Researchers have successfully introduced the missing genes, effectively restoring normal gene expression and significantly improving patients' quality of life. While challenges regarding immune responses to viral vectors and ensuring long-term expression of the therapeutic genes persist, gene addition approaches hold potent therapeutic potential. Gene silencing strategies. Another branch of gene modification is gene silencing, which aims to suppress the expression of specific genes responsible for disease development. One widely used technique is RNA interference, RNAi, which uses small molecules known as small interfering RNAs, siRNAs, to target and degrade disease-causing messenger RNAs, mRNAs. RNAi-based drug therapies have shown remarkable success in treating diseases like hereditary transthyretin-mediated amyloidosis, a rare condition causing organ damage. By effectively silencing the genes responsible for the production of harmful proteins, RNAi-based therapies help curtail the impact of the disease and slow its progression. Advances in clinical trials. The advancement of gene modification techniques has paved the way for numerous successful clinical trials. These trials provide critical insights into the efficacy, safety, and long-term effects of gene therapy interventions. Notable breakthroughs include the approval of Luxturna, a gene therapy for a rare retinal disorder, and Zolgensma, a treatment for spinal muscular atrophy. Furthermore, ongoing clinical trials are exploring the potential of gene therapy in treating a wide range of diseases, including cancer, hemophilia, and Huntington's disease. These trials not only showcase the growing confidence in gene modification techniques but also offer hope for patients suffering from currently incurable conditions. Challenges and ethical considerations. Despite the tremendous promise of gene modification in revolutionizing medicine, numerous challenges and ethical considerations need to be addressed. Additionally, ethical debates surrounding the use of gene modification to enhance human traits or create designer babies warrant thoughtful consideration. With advances in gene modification, and regulations are necessary to ensure responsible and ethical use of this powerful technology. Gene therapy has been a subject of interest and research for many years, but recent advances in gene modification techniques have brought us closer to realizing its immense potential. Gene modification involves manipulating the genetic material of an organism to introduce desired changes. This can be achieved using various techniques, including gene editing, gene addition, and gene silencing. One of the most promising gene modification techniques is CRISPR-Cas9. CRISPR-Cas9 acts as molecular scissors, targeting specific genes and introducing modifications. The simplicity, cost-effectiveness, and high efficiency of CRISPR-Cas9 have sparked immense excitement in the scientific community. With this technique, the potential to correct genetic defects associated with diseases like cystic fibrosis or sickle cell anemia is within reach. Another approach to gene modification is gene addition, where healthy genes are introduced into a patient's cells to compensate for defective or missing ones. Viral vectors, such as adeno-associated viruses, are commonly used to deliver the therapeutic genes into the target cells. Gene addition approaches have shown promise in treating genetic disorders such as spinal muscular atrophy and hemophilia. Gene silencing is yet another branch of gene modification, aiming to suppress the expression of specific disease-causing genes. RNA interference, RNAi, is one widely used technique for gene silencing. It utilizes small interfering RNAs, siRNAs, to target and degrade disease-causing messenger RNAs, mRNAs. RNA-based drug therapies have shown success in treating conditions like hereditary trans-uretin-mediated amyloidosis. Advances in gene modification techniques have paved the way for numerous successful clinical trials. These trials provide crucial insights into the efficacy, safety, and long-term effects of gene therapy interventions. Examples of breakthroughs include the approval of Luxturna for a rare retinal disorder and Zolgensma for spinal muscular atrophy. Ongoing clinical trials are exploring the potential of gene therapy in treating cancer, hemophilia, and Huntington's disease, offering hope to patients suffering from currently incurable conditions. However, along with the promise, there are also challenges and ethical considerations that need to be addressed. Ensuring the safety and accuracy of gene modifications, addressing the potential for unintended consequences, and ensuring accessibility to gene therapies for marginalized communities are key issues. Ethical debates surrounding the use of gene modification for enhancing human traits are creating designer babies require thoughtful consideration and stringent regulations. Gene therapy is an evolving field that has shown remarkable advancements in recent years. Through gene modification techniques such as gene editing, gene addition, and gene silencing, scientists are unlocking the potential of gene therapy to target the root causes of genetic diseases. One of the most promising techniques is CRISPR-Cas9, which acts as molecular scissors to precisely edit genes. His technique brings hope for correcting genetic defects associated with diseases like cystic fibrosis and sickle cell anemia. Gene addition approaches have also shown great promise, where healthy genes are introduced into patients' cells to compensate for defective or missing ones. Viral vectors, like adeno-associated virus, are commonly used for delivering therapeutic genes. This method has successfully restored normal gene expression in disorders such as spinal muscular atrophy and hemophilia. Another significant advancement in gene therapy is gene silencing, which aims to suppress the expression of disease-causing genes. RNA interference, RNAi, is a widely used technique in gene silencing. Using small molecules called small interfering RNAs, siRNAs, to degrade disease-causing messenger RNAs, mRNAs. RNAi-based therapies have shown success in treating hereditary transthyretin-mediated amyloidosis.